every physical feature, from eye color to blood type. Like pearls strung along a DNA necklace, genes can be of different lengths, ranging from a few hundred nucleotides for the smallest genes to a few million nucleotides for the longest. A gene's A, C, G, and T nucleotides provide the instructions to our body about how to construct its different proteins. Proteins, of course, are macromolecules that play innumerable important functions and are the workhorses of our bodies. A protein like collagen plays a structural role in making our bones, ligaments, and tendons strong. The protein hemoglobin in our red blood cells helps transport oxygen and carbon dioxide in and out of our tissues, and the protein fibrin is essential for normal blood clotting. Proteins themselves are actually composed of chains of little building block molecules called amino acids, of which we have 20 different types, and each different protein has a unique chain of these amino acids. The type and order of DNA nucleotides in a gene provides the instructions on how to put these amino acids together. Perhaps surprisingly, however, genes make up only about 1.5% of the entire human genome. This means that only a very small fraction of our vast genome is actually coding for the proteins that carry out important structural and functional roles that make our body work. The remainder of the genome was long considered to be almost entirely junk DNA, that is, non-functional, but more recent studies have estimated that as much as 4% to 12% of this remaining DNA also has important functions and perhaps plays important roles in controlling the expression of genes. For example, many short elements within the genome have been identified that can turn genes on and off, or can increase and decrease the amounts of protein genes produce. These gene switches are responsible for activating different sets of genes in different anatomical regions of our body, which helps to explain why our brains have very different characteristics from our livers. The sequence of a genome is determined through a complex laboratory process using increasingly efficient technologies to resolve the precise order of the nucleotide bases that pair together to form the steps of the ladder within our DNA. Since a human genome is so large, its sequence needs to be deciphered in small segments, which are then assembled together using sophisticated computer programs to produce the entire genomic sequence. One significant finding from initial analyses of our genome is that the number of genes it contains is far fewer than we had long believed. Instead of the 50,000 to 100,000 genes suggested in genetics textbooks as recently as 15 years ago, the human genome is now considered to be made up of approximately 21,000 genes. A fresh look at old questions about our past. But even as old mysteries are finally solved, the new DNA evidence leads us forward by causing us to revisit old and persistent questions about our evolutionary origins, as well as raising important new ones, many of which we would never have thought to ask. One of the greatest events in all of evolution from a human-centric standpoint is the one that led to the divergence of the human lineage from our great ape cousins, the chimpanzees, gorillas, and orangutans. Within these pages, I'll tell the story of the genetic quest, from small stretches of DNA to entire genomes, to trace our past to the origin of our lineage and find our closest ape relative. We now know, of course, that chimpanzees and their close cousins, the bonobos, are our closest living relatives. Along the way, we'll discover some surprising aspects of our genome that show our deep evolutionary connectedness with all the apes, not just chimpanzees. These studies reveal to us that our genome is like a patchwork quilt, with new pieces added over the course of generations.